get ready to pay more for crabs.
Yeah, and this is not because of inflation or tariffs, anything like that. It simply is an issue with the crab population.
For decades, the Chesapeake Bay, one of America's richest waterways, was slowly losing its life. Oxygen levels in the water were dropping. Fish populations were collapsing. Entire communities that depended on the bay were struggling to survive. Scientists tried restoration projects, stricter regulations, and habitat rebuilding, but none of the methods seemed to work. So one research team proposed something different. Instead of waiting for perfect conditions, why not restart the system itself? They released 10,000 crabs into a struggling river. At first, the idea sounded absurd, but once the crabs began feeding, stirring sediment, and reshaping the food web, the results surprised everyone. So how did this method work? And is this the only way to save a collapsing ecosystem? Let's dive in. The dead zone. For centuries, the Chesapeake Bay was one of the richest waterways on Earth, stretching roughly 200 miles and covering about 4,500 square miles. It supported entire communities that depended on seafood for both food and income. Watermen once harvested close to 20 million bushels of oysters each year. Blue crabs filled traps by the millions. Underwater grass meadows provided shelter for young fish, crabs, and countless other species. The bay was not just water. It was a working engine of life. But after World War II, the region began to change. Industrial farming expanded across states that feed into the bay. Farmers used large amounts of fertilizer rich in nitrogen and phosphorus. Rain washed the extra nutrients into more than 150 rivers and streams that drain into the bay. At the same time, growing cities sent wastewater, oil runoff, and sediment into the same system. These nutrients fueled massive algae blooms. When the algae died, they sank and decomposed, a process that consumed oxygen from the water. In many summer months, oxygen levels near the bottom fell below 2 milligrams per liter, too low for most fish and crabs to survive. Scientists began calling these areas dead zones. By the early 2000s, dead zones appeared almost every year. Oyster harvests crashed to a fraction of historic levels, and blue crab numbers dropped sharply. The bay that once fed millions had become a place where life struggled to breathe. And just when it seemed the decline might be permanent, one scientist prepared to test an idea that sounded completely backward. One that would challenge everything experts thought they knew about saving a dying ecosystem. The crazy plan. But cleaning the water alone would not be fast enough. The real turning point came from a scientist who believed the solution was already alive, waiting on the sidelines. Instead of asking how to remove every pollutant first, he asked a different question. What if the missing animals themselves were the key to restarting the system? In 2012, marine ecologist Anson Hines from the Smithsonian Environmental Research Center proposed an idea that many experts first called reckless. Instead of waiting for perfect water conditions, his team would release thousands of juvenile blue crabs directly into polluted water. The goal was not just survival. The goal was to rebuild an entire food web from the bottom up. Blue crabs are not passive animals. They are aggressive scavengers and hunters. They also dig into sediment, which helps move oxygen into the riverbed. Hines believed that if enough healthy crabs were placed in the right spot, they could begin cleaning the ecosystem naturally. These were not tiny hatchlings. The crabs were raised in hatcheries until they reached a stronger late juvenile stage, past their most fragile early life phase. Each crab was measured and tagged so researchers could track survival after release. The chosen test site was the Lafayette River, a polluted tributary with the right mix of salinity and habitat. Release animals into bad water and hope they fix it. It sounded weird, but this risky plan was about to face its first real test. on a humid summer morning when thousands of claws would disappear beneath the surface. Release day. On that humid summer morning, the risky plan finally left the lab and met the real world. Boats eased into the brown-green waters of the Lafayette River, carrying thousands of live blue crabs stacked in aerated bins. For years, the idea had existed only in papers, tanks, and late-night debates. Now it came down to one simple question. Once those crabs hit the bottom, would they live? The team worked quickly because they had to. Summer heat and handling stress can kill crabs in hours. Each crate was lifted, tipped, and emptied over the side. About 10,000 late-stage juvenile crabs were released that day. Not randomly, but in carefully chosen spots where the salt levels were right and small patches of underwater grass still survived. Those grass beds mattered. They offered shelter, and shelter meant a fighting chance. Waiting was the hardest part. The same low oxygen that had damaged the river for years could wipe out the experiment in days. If the crabs sank into water that could not support them, no one would know until it was too late. 3 days later, the researchers returned. They lowered baited crab pots into the exact release areas and marked each location. When they hauled up the first trap, the boat fell silent. Inside the wire cage, bright blue claws thrashed. The tags confirmed it. These were the hatchery crabs, alive, alert, pushing against the mesh as if offended by the interruption. It was the first real sign that the risk might pay off. Over the following weeks, more tagged crabs showed up in traps and on underwater cameras. They were not drifting downstream. They were staying put. They burrowed into the sediment. They defended small patches of riverbed. They were behaving like wild crabs, not lab animals released by mistake. But staying alive was only step one. The scientists were not trying to add crabs for a few weeks. They needed proof of something bigger, the chain reaction. What truly mattered next was whether those crabs could change the river around them, because in nature, one species rarely acts alone. Every animal is connected to many others through food, shelter, and competition. If the crabs were going to make a difference, the signs would start appearing in places no one expected. Within months of the 2012 release in the Lafayette River, researchers began recording shifts beyond crab counts. Blue crabs are bottom feeders and active hunters. They eat small clams, worms, and other creatures that stir up sediment. By controlling these populations, they changed how the riverbed functioned. Clam numbers began to rise in areas where crabs kept their predators in check. This mattered because clams are natural filters. A single clam can clean liters of water each day by removing particles and algae. As clam beds expanded, the water slowly became clearer. Clearer water allowed more sunlight to reach the bottom. That sunlight fed the underwater grass. Surveys showed seagrass coverage in test areas increasing from roughly 15% of historic levels to more than 20% within a year. These grass beds provide shelter for juvenile fish and crabs, protecting them from predators and strong currents. Bigger animals soon followed the food. The big lesson: the chain reaction did more than improve one river. It challenged a belief that had shaped environmental science for decades. For years, the main strategy was simple: reduce pollution, install expensive treatment systems, and wait for nature to recover on its own. The results were slow and often disappointing. The experiment in the Chesapeake Bay showed another path. By 2015, programs in Virginia expanded crab releases to more sites. Over the next several years, hundreds of thousands of juvenile crabs were added across different tributaries. Monitoring showed consistent trends in restored areas. Dissolved oxygen improved near the bottom. Underwater grass beds expanded. Fish diversity increased compared to nearby untreated waters. The lesson was clear and practical. Certain species do more than live in an ecosystem. They help run it. When those key species disappear, the system can stall, even if pollution levels slowly improve. Reintroducing large populations can restart natural processes such as filtration, nutrient recycling, and habitat formation far faster than machines alone. This does not mean pollution no longer matters. Runoff and waste still need to be controlled. The Chesapeake Bay was a living system built on connections between animals, plants, and tiny creatures we rarely notice. For years, many believed a place this damaged could only recover slowly, if at all. But this project showed that when a key species returns, nature can begin to repair itself in ways no machine can truly replicate. So, what do you think? Should scientists try reintroducing key species like blue crabs into other damaged rivers and bays to help nature rebuild itself? Drop your thoughts about this in the comments below. If you liked this video, ensure you give it a thumbs up. Also, consider subscribing to the channel if you haven't already, because we will be uploading more crazy stories like this one in the coming weeks. Until then, see ya.